Thank you for giving me the opportunity to present our data on the neurogenetics disease study in Mali. So, as always, I would like to start by saying that Africa is the largest continent in the world with a very diverse population. It is also the house of more than 30% of the world's identified resources. But still, if we project Africa's place in terms of innovation, it holds a small place, so shown on the map here. This is because not much research is ongoing on the continent, especially genetic diseases. Hereditary neurological disorders are mostly incurable and debilitating with a high burden of disease, like the disability adjusted life years, and also social factors like stigma and limited resources make it worse. They are clinically and genetically heterogeneous. Several clinical entities were characterized in the Caucasian population, and today more than hundreds of genes have been identified. But still, many of them showing a Mendelian pattern are with no molecular diagnostic. But Africa, and Mali in particular, represents a unique environment to do gene finding studies. First, because of the consanguineous and intra-ethnic marriages, and also the high fertility rate beyond 6 live births per woman, for example, in Mali. Also, the population is very diverse and still understudied. That's how before 2007, all work done on neurogenetics in Mali was about the clinical characterization of families with neurogenetic disorders, or genetic studies on Malian families abroad or from the diaspora. That's why in 2007, we undertook a pilot study including 27 families and found new clinical entities that are caused by, some of them caused by novel variants and genes we have published earlier. But many families still remain with negative testing for all known clinically relevant genes. That's how we stipulated that the Malian population might bear clinical entities or variants that might be due to genetic defect or other environmental factors. So we put forward an H3 Africa application with the aim to characterize families with hereditary neurological disorders, identify mutation causing these diseases, explore them in cell culture models or animal models, but also train faculty members and students in genetics and molecular biology. To date, we have more than 300 families enrolled, with more than 1,000 participants. More than 500 of them are patients. DNA has been collected, and for some of them, we have established the IPS and neuronal cell lines. Of note, not all of them have neurological disorders. And also, not all of them are from Mali. As you can see, many of our patients also come from neighboring countries. This is raising the need of doing rare disease exploration in this part of Africa. So I would like to start by giving some of the major phenotypes we have found here. Here I'm showing ataxia characterized by slowly progressive abnormal gait, as you can see on the video here, and dysarthria. We We have seen dominant cases, more than 30 families, and recessive cases. Several of the recessive cases came back negative for all clinically relevant genes. I would like to highlight here one family with Friedreich's ataxia. We published earlier this year. Friedreich's ataxia is a very rare recessive ataxia that is still to be found in the population with Black ancestry. The second major phenotype is myopathy, characterized by proximal weakness, and waddling gait, high CK levels. We have found 59 families, and some of them we have established the molecular diagnosis, and highlighted here one novel variant on the GNE gene, which causes a very rare distal myopathy in the world, but also one potential novel gene for congenital myopathy we are validating. The third major phenotype is Charcot-Marie-Tooth, which is the most common inherited peripheral neuropathy characterized with distal sensory and motor involvement. You can see here the steppage gait, the abnormal steppage gait, but also on the hand you can see this difficulty to raise the hand. More than 50 genes have been associated with CMT. We have enrolled 31 families. We screened some of them with some of the novel mutations, but also some of them were negative. We recently identified a novel mutation in this gene that was recently published in Brain for a mutation in 3 families with the same background. So this highlights also the difficulties African researchers have to put their data out, because this was submitted in 2020, but we had our data since 2019. So functional analysis has been slow. With this Charcot-Marie-Tooth, we have a BAG3 mutation, novel variant also. Only 5 families were described worldwide. It's a polyneuropathy and cardiomyopathy. You can see here the index patient who has a heterozygote mutation in this gene. Another very rare subtype of CMT, like one single family described worldwide. Most patients in that family were asymptomatic. However, the Malian family has distal weakness and atrophy and sensory loss, but also they have hearing impairment. We have found in the index family a novel variant in this gene shown here on the chromatogram. Another major clinical phenotype is familial epilepsy, and here we are presenting a family with cerebral folate deficiency. Only few families were described worldwide, and recently one in Gambia, which is not a neighboring country, but not a country with the same ethnic background. So we have found a novel variant in the 4-1 gene. This was not found in the database, but fortunately, it has been shown that a high dose of folinic acid, which is available on the market, can ameliorate and even prevent further degeneration in this disease. So our family was put on this medicine with an improvement, with the improvement in the symptoms. The last major group we want to present here is spastic paraplegia, so characterized by spasticity in lower limbs and with some minor weaknesses. 30 families were collected, many of them being recessive, some dominant, and 6 of them were classified as sporadic because of no other pattern going toward one of the inheritance patterns. Molecular genetic was done in 4 families, in which we found novel variants in SPG 10, SPG 11, and also some rare variants seen in the Jewish and Arabic population. Interestingly, 5 of them were negative for all candidate genes or panel testing. One of them is a large consanguineous family with spasticity and seizure and major and mental retardation. So you can see here, you can see here how this young is disabled with walking difficulty, but also some abnormality in the feet. So here we have done the first surgery that allowed him to be able to put this feet flat. With this family, we have done HTLV1, that causes tropical spastic paraplegia. It was negative. Vitamin E level were normal. EEG was abnormal, and MRI has shown a thin corpus callosum that with a characteristic, which is characteristic of spastic paraplegia, but also cortical and cerebral atrophy that is shown here on the these MRI images. Candidate gene and SPG panel testing was negative, but whole exome sequencing has identified a novel variant in the AP2A gene. The AP2A gene is a member of an AP complex, some of which were involved in other SPGs, like the SPG4 and the AP4 and the AP5. So when we collected fibroblast cell lines in the patient and the mother and other family members, they were differentiated to cortical neurons. And when we checked protein levels, we could see in the phosphorylated cells, we can see that here you can see that the control has 2 bands, and the mother has 1 band and an upper faint band. However, both patients do not have bands. When we did, when we removed phosphorylation, and we can see that the control has 4 bands. The mother has also 4 bands, but the 2 upper bands are very faint bands, and the 2 patients have only 2 bands. So this work was done by our fellow at NIH, and we could clearly see from this Western blot that there is a difference between the wild type and and the mutated cells. So she went on to do immunofluorescence staining with claritin, and you can see from the patients and the heterozygote mother and the control, you can see that the merging is different from the patient and the mother, and here the control. This all shows that this mutation is really damaging. It was expected to be damaging with in silico prediction, but also in cell culture models. So this is really a tiny part of what we could find in our court, and also a tiny part of what could be found in Africa. This study and others who presented in this meeting and in previous meeting have shown that Africa really harbors a high genetic diversity and genetic variability that these recent studies might find and help in developing, advancing the understanding of the diseases, but also going forward with precision medicine or therapeutic findings. So we do not do only we do not see only patients and characterize them molecularly, but we do community engagements through workshops and lectures with high school students or university students, but also we go to the countryside to talk with the population or hold radio talk shows to explain these diseases to patients and the population in order to get them engaged in our studies. We do train students. Here you can see our students at NIH during one of my visits, and a student performing her sequencing data analysis, showing me how she's doing it. It's true that we go to nice meetings here, a cruise trip in Washington, DC, a very nice place. But also we do go to difficult places, as you can see here in the countryside, in a really inclement roads, or sometimes working late at night with only the flashlight, or being stuck in the sand in the middle of nowhere with all the security concerns we can see there. So this is how we get advanced in research in Africa, going to difficult places, talking to patients, and getting them involved in our study. So we were able to recruit a large cohort of hereditary neurological disorders. We have improved the clinical characterization of these diseases with some novel variants or novel genes, but we still need to develop research infrastructures and resources in order, and also improve data analysis to close the gap with the other parts of the world. And it is only with that we can improve research and development in Africa and increase this part and increase Africa so that we can clearly see its shape. So I would like to end by acknowledging God, my parents, and all the participating families, to acknowledge the funders and our university, and a special thanks to all the students and the other team members who make this work progress with their enthusiasm and their availability. And thank you for listening, and please do ask questions. Thank you very much.